Good morning, students. In today's class we are going to see about the experiment qualitative analysis of the unknown sugar called carbohydrates, glucose. So for any carbohydrate we have to do systematically the following test. First we have to go on for the Molish test. So Molish test is the one which shows whether the carbohydrate is present in our sample. So for that I am taking the solution, that is sugar solution. This is nothing but glucose. So I am taking 2 ml of glucose solution, and to this I am adding 3 drops of alpha naphthol. 3 drops of alpha naphthol. Then I am mixing it well, and to this I am going to add the concentrated sulfuric acids, that to 2 ml along the sides of the tube. You have to add it slowly because it's an acid, that too concentrated sulfuric acid. So what happened? The observation is, you can see that you can expect a violet color ring at the junction of the 2 layers. It slowly forms. You should not disturb it. At the upper and between the lower part, generally a violet ring is observed. So this is a violet ring is slowly it forms. So like this you can see, expect a violet color ring at the junction of the 2 layers, 2 liquids. So this test is specific for the conformation of carbohydrate. So this is called Molish test. The next test we are going to see the carbohydrate, whether it's a reducing sugar or non-reducing sugars. We have 2 types of sugars, reducing and non-reducing. Reducing means the free functional group called aldehyde or keto group will be not involved in the bond formation. So those types of sugars are called reducing sugars. Example, glucose, fructose, maltose and lactose, all these are reducing sugar. But sucrose is not a reducing sugar because the free aldehyde and free keto group is not exposed and it is involved in the bond formation. So what happened? This test is going to identify whether the given sugar is a reducing or non-reducing. So for this I am taking again a solution of sugar solution. See this, I have taken 1 ml, and to this I am going to add 1.5 to 2 ml of the Benedict's reagents. So Benedict's reagents, why we are adding means it's a one, you can see, expect this is a copper sulfate solution, that's why it's a blue color. This copper sulfate solution, whenever the carbohydrate, glucose or fructose is there or free reducing sugar is there, that undergoes as a, in the presence of the alkaline medium, what happened? It is exposed to enediol form, that means a double bond and alcohol is formed. This is acting as a very good reductive reagent or reducing agent that reduces the cupric ion to cuprous oxide. This is 3 minutes you have to heat it. Burner for at least 2 to 3 minutes. So you can see that blue color is changing to green color. So that shows the concentration of glucose in the solution. If it is 0.5, so see that it is changing to green color. So it has little bit of the glucose molecules getting colored, converted, and convert this cupric ion to cuprous oxide. So and you can see yellow, orange, and finally you can expect the brick red color. This is the brick red color. So this almost all the copper which is present in the Benedict solution converted from cupric ions to cuprous oxide. That's why we are getting the brick red color solution. This shows the presence of reducing sugar. So glucose is a reducing sugar. So our sugar should be a reducing sugar. So this will confirm the test whether we have a reducing or non-reducing. Fourth reagent, which is in blue color, now it been changing to blue to reddish color, red precipitate. That is itself shows that again this is a presence of monosaccharide. So when you heat it exactly to the 3 minutes, you can see the changes from blue color to the light red precipitate. So that is confirming the presence of monosaccharide. If it's still in the blue color itself, you can say that it is a disaccharide. It is not a monosaccharide. So the confirmation is for the presence of mono or disaccharide, which is given as an unknown. The next test we are going to see is Saliva test. Saliva test is very specific for the keto sugars. Keto means here it refers any fructose or even sucrose when hydrolyzed will give a color positive for the Saliva test. So here again I am taking 1.5 ml of sugar solutions and then Saliva reagent for 1.5 to 2 ml. And these two you have to apply the heating condition. So when you heat this, and what is the specific point you have to remember here is you have to heat exactly 30 seconds. You have to exactly heat it to 30 seconds. So in the Saliva test, again the sugar which are having a free aldehyde or keto groups, here keto group specifically undergoes the furfural derivative and reacts with the resorcinol to give a cherry red color. So 30 seconds over and you are not able to see any color change because this is the aldo sugar, that is glucose. So this is negative for the Saliva test for the glucose. If it is a fructose, you will be getting a cherry red color that shows the positive test for the keto sugar. So if for my today's test, this shows a negative. That shows that there is no keto sugar, only aldo sugar is there. So it confirms the given sugar is found to be a glucose. So these are the ways you have to, these are the thing you have to be identify any sugars. So first test is smallish. You can see here violet ring at the junction of two layers. So it shows the presence of carbohydrate, and this either the sugar is a reducing or non-reducing. Bandage test will answer. You will be getting a brick red color that shows the presence of reducing sugar. And Barfoot's test is going to identify whether it's a di or monosaccharide. Since our glucose is a monosaccharide, so we are getting a red precipitate for the Barfoot's test. So it confirms the presence of monosaccharide. And last one is Saliva test. Saliva test doesn't change any cherry red color formation. Shows the presence of aldo sugar, that is glucose. So this is the systematic way of doing the practical experiment for the known sugar called carbohydrate. So that too today we have done only glucose. So these are the way test you have to proceed, and from the confirmation you can say that what is the sugar given for that in that particular day. So in today's we have seen only glucose is given. So that is confirmed with the Saliva test, and it shows a negative color formation, that is no cherry red color is formed. So we confirm that today's sugar what we get in our experiment is glucose.